This is a microscopic picture of cells that line the respiratory system. The cells are lined with microscopic hairs called cilia that serve as a protective system for the lungs. The cilia are active in water, and the CFTR protein precisely regulates the liquid layer. CFTR is a protein which is found on the cell membrane. Phosphorylation of the regulatory site causes ATP binding at two sites, which opens up the chloride channel, and chloride passes out of the cell. The chloride has a negative electrical charge, and when it exists, it attracts the sodium in an electric voltage drop, and sodium chloride is the salt that attracts the water in an osmotic drop. On the cell wall is a sodium channel that transports the sodium into the cell. The chloride passes in the electrical drop and the water in the osmotic drop. The CFTR channel regulates the activity of the sodium channel. When the CFTR is activated, there is a delay in the sodium channel which leads to hydration. When there is a decrease in CFTR activity, there is a decrease in the delay of the sodium channel and liquid is absorbed. When CFTR does not function properly, the aqueous layer which covers the respiratory airway dries up, as can be seen in the following movies.
The CFTR gene is located on the long arm of chromosome 7. Under normal circumstances, the DNA is read and transcribed to messenger RNA, during which the introns are spliced out. The messenger RNA strand exits the nucleus of the cell and travels to the ribosome, where it is translated to the immature CFTR protein product. In the endoplasmic reticulum, or ER, other proteins called chaperones help to fold the protein. CFTR is then transferred to the Golgi apparatus. From the Golgi, transport vesicles are created and the CFTR protein is transported to the cell surface membrane. This process is also known as trafficking of CFTR. The cystic fibrosis transmembrane conductance regulator, or CFTR protein, is located in the apical membrane of cells lining the airways and also can be found in other areas of the body, including the sweat glands, intestinal, reproductive, hepatic, and renal epithelia. The CFTR protein facilitates the transport of chloride into and out of cells. CFTR is a complex glycoprotein composed of 1,480 amino acids. CFTR has a regulatory domain that contains phosphorylation sites and connects the proteins to halves. The CFTR protein has two nucleotide binding domains, NBD1 and NBD2, and two transmembrane domains, TMD1 and TMD2. The movement or conductance of chloride across the CFTR protein requires phosphorylation of the regulatory domain by cyclic AMP. In addition, binding and hydrolysis of ATP are required for the opening and closing of the CFTR channel. This process is also known as gating of CFTR. In the sweat glands, CFTR is a major mediator for chloride absorption. Under normal conditions, chloride and sodium ions are reabsorbed via CFTR and sodium channels, respectively. In cystic fibrosis, chloride reabsorption is diminished due to impaired or absent CFTR protein, and sodium uptake is also reduced, resulting in increased sodium chloride in the sweat. In the normal lung, the airway surface liquid, or ASL, supports an environment in which cilia can beat freely and clear mucus that has trapped inhaled particles. Active secretion of chloride via the CFTR channel and absorption of sodium are necessary to regulate the volume of the ASL. In cystic fibrosis, chloride secretion in the airway epithelia via CFTR is either absent or impaired, and sodium is hyperabsorbed. These defects in ion transport lead to depletion of the ASL, which results in decreased mucus clearance, thick secretions, and obstruction of the airways. Bacterial colonization due to the lack of clearance of thick, sticky mucus from the airways is a contributor to the progressive lung disease in cystic fibrosis. The patient's inflammatory response to the infection and the bacterial products destroy the airway wall and result in bronchiectasis. Bacterial colonization also increases the number of glands and secretory cells in the airway, which increases mucus secretion and obstruction of the airways.
A decrease in CFTR function causes a decrease in aqueous secretion in the epithelial cells that line the exocrine system. And in the lungs, it causes the layer that lines the cells to dry up. The mucus becomes sticky. The ciliary cleansing system is impaired, which causes repeated infections in the respiratory airways. The infections cause an inflammatory reaction. Especially in cystic fibrosis, the reaction is extreme and exaggerated, and that in itself causes damage. There are many factors that cause the extreme inflammatory reaction, and they can be subdivided into external, intercell, and factors connected with the faulty CFTR. These factors all cause damage as a result of pressure in the cells. The inflammatory reaction is mainly neutrophilic, that secrete enzymes that cause damage in cells of the respiratory system. The dead cells release DNA, which in itself increases the stickiness of the mucus in the respiratory system. Faulty CFTR function, reduction in ciliary cleansing, sticky mucus, and the disruption of the inflammatory reaction all cause damage to the respiratory system, which in itself exacerbates each one of the causes, leading to extensive damage to the respiratory system. No structural fault in the lungs can be detected in a child born with cystic fibrosis. But with time, as a result of recurrent infections, bronchiectasis develops. Bronchiectasis is the widening of the bronchi, as can be seen here in the lung tissue and in the CT scan. When cystic fibrosis was first described in the 1940s, infants died within the first year of life from malnutrition and pulmonary infections. With a better understanding of the development of the disease, treatments were started including antibiotics, physiotherapy, inhalations to soften the mucus and to increase mucus passage, and dietary enrichment. All these resulted in a significant life expectancy for CF patients. Nowadays, the median survival is close to 40 to 50 years. This slide shows that lung functions at each age are higher over time. This demonstrates the effectiveness of treatment and the increased life expectancy and quality of life of the patients. New treatments that aim to correct the genetic mutation are being developed and marketed. Most efforts are focused on treating the various systems in which the different mutations lead to a reduction in the CF protein function.
Several types of compounds are under investigation that specifically target dysfunctional CFTR. Three major types are suppressors of premature stop codons, CFTR correctors, and CFTR potentiators. Premature stop codon mutations prevent full translation of messenger RNA, resulting in truncated CFTR, which is not functional and never reaches the cell surface. To address this problem, agents are under investigation that suppress the premature stop codon, allowing translation of a full-length CFTR protein that then reaches the cell surface, where it may be able to transport chloride to the airway lumen. Other CFTR mutations affect post-translational folding and transport of the protein, also known as trafficking. The result is that only a small quantity of CFTR protein reaches the cell surface. Investigational agents called CFTR correctors promote folding of CFTR, resulting in its exit from the endoplasmic reticulum and transport to the cell surface. At the cell surface, CFTR may then be able to transport chloride to the airway lumen. With other CFTR mutations, although the CFTR protein reaches the cell surface and is present in normal amounts, it fails to transport chloride due to impaired opening of the channel, also known as gating. Investigational agents called CFTR potentiators aim to increase the amount of chloride secreted by the airway cell by improving the opening of the CFTR protein.